Hello, everyone, and welcome to today's webinar, CliniMACS Prodigy Adherent Cell Culture System, Automated Large-Scale and GMP-Compliant Manufacturing of Various Stem Cells and Their Derivatives. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more about our sponsor, please visit www.miltenyibiotec.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Sebastian Knöbel, the Manager of Research and Development at Miltenyi Biotec, and Chao Sheng, the Product Manager of Regenerative Medicine at Miltenyi Biotec. For the complete biographies on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Okay, hello. Welcome to our presentation. So we are actually very glad that we have this great opportunity to present to you our latest technologies for automated GMP-compliant and scalable stem cell manufacturing. My name is Chao Sheng. I'm the product manager for Regenerative Medicine portfolio at Miltenyi Biotec, and I hope that you will find our presentation helpful for your project. So first, this is our presentation disclaimer. Then at first, I would like to show you a few key facts about Miltenyi Biotec. Our company was founded in 1989, and after 30 years growth, today the Miltenyi family holds 2,500 members from 44 different nations who are committed to empowering scientific discovery and also impacting human lives. Every year, there are more than 55,000 cell therapy procedures are performed using our products, and more than 2,500 patients are treated with cellular products using our technologies. In the last 30 years, we have gained extensive experiences in cell manufacturing. We opened our GMP manufacturing facility in Tettau, Germany in 2002, and in 2012, we launched the CliniMACS Prodigy as a next-generation platform for commercialized cell manufacturing. Based on the CliniMACS Prodigy platform, in 2016, we released our T-cell transduction system for CAR T-cell manufacturing, and in 2017, we opened our Miltenyi Biotec cell factory as a grand GMP cell manufacturing facility. Today, there are more than 15,000 products in our portfolio supporting scientists and patients from benchtop to bedside. In this presentation, we will focus on the cell processing part, especially our cutting-edge technology for automated and scalable stem cell manufacturing. Sitting next to me is Dr. Sebastian Knöbel, project leader of our brand new CliniMACS Prodigy Adherent Cell Culture System. This automated and closed system is dedicated to addressing the challenges in GMP-compliant and large-scale manufacturing of various adherent cell types, such as mesenchymal stem cells, pluripotent stem cells, and their derivatives. Sebastian will share with you our solutions to automate, to standardize, and to scale up the manufacturing processes, and at the same time to reduce the production costs and make cellular products more affordable for patients. More detailed information about the CliniMACS Prodigy Adherent Cell Culture System can also be found in the links to the Miltenyi website. Without further ado, so now I would like to give the time to Sebastian.
Thank you for the nice introduction and also welcome to our talk from my side. As you might know, GMP manufacturing of a cellular product is a very complex endeavor. As depicted in the simplified process scheme, it involves a multitude of devices in sophisticated cleanroom facilities, manual handling steps, and skilled personnel. So how to make a cellular product in a scalable and affordable manner? One of the biggest challenges of GMP cell manufacturing is the complexity. And this significantly drives up the costs. And at the same time, it limits the manufacturing scale and consistency. But how to make these very complex cell manufacturing processes standardized, scalable, but at the same time affordable for patients? Our solution is to integrate all these various steps into one automated device. And here comes our CliniMACS Prodigy platform, designed for complex cell product manufacturing. I would briefly like to go through the CliniMACS Prodigy Adherent cell culture system with you, including the hardware and software. Let's have a closer look at the hardware. This is the functionally integrated and closed CliniMACS Prodigy instrument. Together with a disposable single-use tubing set and other accessories, the Prodigy system allows automated cell processing from starting materials to final products. Upon your request, it can perform sample preparation, density gradient centrifugation, cell washing, medium change, magnetic cell separation, cell stimulation, genetic modification, but also final product formulation. Now, what are the main features of the closed system? We use sterile single-use tubing sets in which all processes take place. So cells are never in contact with the device itself at any time. So we provide single-use sterile filters at the input ports, and the starting material is connected via sterile docking. Buffer and media bags can be sterile welded on and off during the process, and the final product is retrieved by sterile unwelding of the target cell bag. QC sample pouches are also provided and part of the tubing set, and can be retrieved during the process for in-process controls. Cell cultivation can be performed in the centricult unit and external cell culture vessels. There is a CliniMACS Prodigy chamber that lies inside of the centricult unit, or so-called CCU. The CCU is the heart for cell manufacturing. It supports automated adherent and non-adherent cell cultivation. In this chamber, there is a temperature control. You have CO2 and air and N2 control, and you can visually examine the cells through a microscope. Additionally, the external cell culture vessel allows scalable manufacturing, since they can be connected to the tubing set via sterile welding and enlarge the surface that is available. Key piece of equipment is the tubing set. The TS730 has been specifically designed for adherent cell culture system to meet the requirements for cell manufacturing in terms of handling and usability. This new GMP tubing set provides additional tubing connections and increased tubing length to allow comfortable operator interaction during the process. That is, for instance, when connecting or disconnecting bags. With this, I'd like to shift your attention to the software application. To the right, you see a menu screen that gives an indication of the different modules that we provide. For manufacturing of adherent cell types like pluripotent cells, neural cells, cardiomyocytes, and MSC, we have developed this specific new software. We call it the Adherent Cell Culture System. The biggest challenge here is that there are so many different types of adherent cells that we really have to make sure that our system is flexible enough to meet all these demands. That is why we modularized individual cell culture steps in the system. For example, in the starting screen, you can find modules like density gradient centrifugation, coating, media change, culture, and other relevant steps. And the adherent cell culture system allows free combination of different modules, thus offers a high degree of flexibility for various cell culture applications. Coming back to the process scheme that I introduced to you earlier, you may notice that all process steps are reflected by the different modules in the ACC system. And within these modules, you can choose between the different cultivation vessels, the CCU or ECV. And you can also set up important cultivation parameters upon your request. For example, in the coating module, you can set up the coating temperature, the coating time, and coating solution volume according to your manual protocol. You will have an overview in the summary page, and you can also reset if you decide to change any parameters. The idea is combining our GMP-compliant CliniMACS Prodigy instrument and tubing set, the highly flexible ACC software, as well as our Max GMP medium, cytokines and small molecules. Our CliniMACS Prodigy platform could support the entire manufacturing process of adherent cell types. With this, I'd like to introduce some of the relevant applications that we have developed on the CliniMACS Prodigy Adherent Cell Culture System. For BSC-related applications, we have developed xeno-free media for high-quality human pluripotent stem cell expansion. First, we have the StemMACS IPS Brew xeno-free media, which is a very cost-effective research-grade media. In this experiment, 2 IPS clones were expanded for 20 passages in the media. We found that cells had very stable doubling times during long-term cultivation. Using StemMACS IPS Brew xeno-free media, human pluripotent stem cells could be cultivated as single cells or as cell clusters. Meanwhile, if we want to translate it into a clinical-grade protocol, we will use the IPS Brew GMP medium, which is a GMP-compliant media manufactured under ISO 13485. The IPS Brew GMP medium is based on the same formulation as the research-grade medium. Performance-wise, they give rise to very similar results. Therefore, you will have a seamless transition from research-grade to GMP-compliant human pluripotent stem cell cultivation. GMP-compliant human PSC manufacturing can be realized in the Prodigy ACC system by using different modules. This graphic scheme gives you an overview of this ACC-based PSC expansion process, and you can find more detailed information such as consumables, hands-on time, scale in the specific application sheet. So let's first have a look at the setup for the pluripotent stem cell expansion. What you see in these experiments is the comparability study between the two systems. And what you can see is that cells have a very similar phenotype. So over a course of 3 passages, they have a very comparable immunophenotype, and they keep the same differentiation potential as they have in the manual system. Concerning the scales, it's worthwhile to mention what kind of scales we have in the different formats that we can provide. We can amplify up to 5 times 10 to the 7 pluripotent stem cells in the CCU, in the chamber, and up to 2 times 10 to the 8 in a 1-layer cell stack. And this means that in a 5-layer cell stack, we can go up to 1 to 10 to the 9 cells, which is for sure cell line dependent. Next, we are wondering if we can use the CliniMACS Prodigy system to differentiate pluripotent cells into specific cell types, for example, midbrain dopaminergic progenitors. This Prodigy-based application has been supported by a prestigious EU consortium, Neurostem Cell Repair, in which we have collaborated with Dr. Marlene Palmer and Dr. Agnete Kirkeby in developing a GMP-compliant protocol for PSC to MSDA progenitor differentiation. We first adapted the protocol in our system using our high-quality cell culture media, cytokines, and small molecules. Then we transferred this manual differentiation protocol to the CliniMACS Prodigy system and developed an automated differentiation protocol, including stringent QC and IPC procedures. Again, this is a process overview, and you can find more details in the dedicated application sheet. We developed specific flow cytometry-based QC panel, which enables determination of the correct identity, purity, and potential tumorigenicity of the resulting MSDA progenitors. We chose the positive markers IAP, FOXA2, OTX2, NKX2-1 to identify the ventral midbrain region of the floor plate. QC assays demonstrated that Prodigy-based differentiation gives rise to a high-purity MSDA progenitor population, which with nearly no contamination of residual pluripotent cells, and untargeted neural cell populations. Rostro-caudal patterning was driven by increasing Kaya 99201 concentration, dorsoventral patterning by increasing human sonic hedgehog concentrations. Expression of PAX6 is indicative of a dorsal phenotype, whereas NKX6.1 and loss of OTX2 expression are associated with a caudal phenotype. SOX1 identifies a dorsal phenotype as well as primitive neuroectoderm. Ki67 was included to monitor the proliferation capacity, and OCT34 served as a marker to identify the residual pluripotent cells. And what you can see by comparing it, that we can clearly identify the ventral midbrain dopaminergic neurons and discriminate them from dorsal forebrain or ventral hindbrain states that could arise in an aberrant differentiation pathway. So next we compared how the different systems correspond. So we compared the manual differentiation procedure and the CliniMACS Prodigy-based differentiation procedure, and we found that both is very comparable. With more than 90% of cells co-expressing the key mesencephalic dopaminergic progenitor marker OTX2, IAP, and FOXA2, nearly no expression of PAX6, SOX1, and OCT4, and decreased expression of Ki67. So let's have a look at the scalability. Starting from 1 million pluripotent cells, after 5 days pre-expansion and 16 days of differentiation, we can get roughly 4 billion high-purity MSTA progenitor cells, which translate to almost 800 patient doses. Therefore, we can realize highly efficient and scalable human pluripotent stem cell differentiation in our CliniMACS Prodigy system. So with this, I'd like to shift to another application, which is the MSC expansion. So it's a very similar case for human MSC manufacturing. First, we have developed the research-grade StemX MSC expansion media and the MSC Brood GMP medium based on the same formulation. And with this, we can now realize GMP-compliant and scalable MSC expansion in the closed and automated system. We also have a dedicated application sheet for this application. What you can easily take from this slide is that after 14 days of expansion, approximately 4 × 10⁸ MSCs can be harvested using the CliniMACS Prodigy system, which is comparable to manual processing. A flow cytometry-based QC assay using our MSC phenotyping kit was performed to confirm the quality of the resulting MSCs. Harvested MSCs met ICT criteria, showing high expression levels of MSC-specific markers, CD73, CD90s, and CD105, and very low expression of non-MSC lineage markers. The Prodigy system could also be your time saver, especially at the DGC step. All in all, the Prodigy system can reduce the hands-on time of for MSC expansion by roughly 70%. So we can also realize the isolation, expansion, and harvest of MSCs in the automated and closed system. We've also extended this workflow to various tissues, including bone marrow, umbilical cord, and adipose tissue in the closed system. We can now harvest clinically relevant numbers of MSCs with high purity from various tissues within 20 days. So all in all, we have the 3 major applications developed. This is pluripotent stem cell expansion, MSTA progenitor differentiation, and MSC expansion. And this allows flexibility for manufacturing of various adherent cell types, since this is not limited to these cell types, and lots more protocols can be translated according to the flexible software. So in the last few minutes, I would like to share with you our ideas how the CliniMACS Prodigy system can actually improve your cell manufacturing. And therefore, I'd like to highlight some key advantages of the system. So actually, the system allows a parallelized cell manufacturing scheme and simplified track transfer and increased user and product safety. Also, it reduces the requirements for clean room and personnel, which in the end means cost reduction. So reduced hands-on time and operator influence will also lead to higher consistency. The Prodigy system has been provided in 31 countries already, and it gives us experience in many different regulatory environments. The technology has been invested in by many drug developers and cell and gene therapy companies. Over 25 trials are underway using the CliniMACS Prodigy system worldwide. In fact, 20% of our customers own more than one instrument, showing the strength of the CliniMACS Prodigy system for commercial scale manufacturing. The protocols and data I just showed can also be found in our application sheets and scientific posters. If you're interested, we could send more detailed information to you. With this, I'd like to come to an end and acknowledge our collaborators and also the projects that have been generously funded by the EU and Germany, and also acknowledge all people who have contributed to the work. And with this, I'd also thank you for your attention and like to take your questions after this talk.
Thank you, Sebastian and Chao, for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is there a limit in the number of media that one can use over the course of PSC expansion and differentiation?
Actually, in principle, there is no limit in the number of media that can be used during expansion and differentiation process. Using the accessories, and that is the connectors, adapters, tube extension, etc., our customers can always connect additional media bags to the tubing set to fit their protocols. For instance, in the ACC-based MESDA progenitor differentiation workflow, we use 4 consecutive media. First, iPS-Brew GMP media, then a neural induction medium, neural proliferation medium, followed by neural differentiation medium. So actually, you could extend this even.
Great, thank you. Your next question is, what are the scales of PSC and MSC in manufacturing in the CliniMACS Prodigy Adherent Cell Culture System?
Using the system, our customers can perform scaled manufacturing of all these cell types, and as also briefly mentioned in the presentation for the pluripotent cells, we can culture up to 5 times 10 to the 7 cells within the chamber. Then it's 200 million cells in a 1-layer cell stack and up to 1 billion cells in a 5-layer cell stack. And if you connect more cell stacks to the system, the pluripotent stem cell expansion scale will be even larger. These cell numbers are, of course, cell line dependent, as I mentioned, and different lines may give rise to slightly different results, but this is actually quite obvious for everyone working in the field. For the MSCs, from 2 to 3 5-layer cell stacks that can be used in parallel, we can harvest roughly 200 million to 800 million MSCs. Roughly within 20 days, depending also on the starting material from various tissues that we can put in, like bone marrow, umbilical cord, adipose tissue. And again, the cell numbers also depend on the quality and quantity of the starting material, as well as the expansion time.
Thank you. Your next question is, do the users need to operate the device under a clean bench to ensure sterility?
No. As we pointed out in the presentation, the Prodigy ACC-based cell manufacturing process is performed in a closed and GMP-compliant tubing set, or in this external culture vessel connected via closed and sterile welding, for instance, using a Terumo welder. And according to our knowledge, also the conic cell stacks are suitable for GMP manufacturing. Therefore, technically, our customers can perform GMP-compliant cell manufacturing in a Class C room, and there is no need for a clean cell culture hood. And so it's the Class C designation actually derives from the European GMP guidelines. This will actually correspond to a 100,000 according to the US designation, or ISO Class 8.
Great, thank you. Your next question is, can you explain why the Prodigy saves time in comparison to manual production?
Yeah, this is because the Prodigy-based manufacturing is automated, although user interactions are still required. But for example, in our ACC-based MSC expansion workflow, the user can choose the density gradient module to isolate mononuclear cells from bone marrow aspirates. And this density gradient centrifugation process is fully automated in the centrifugal unit. This is a major time saver for the entire workflow. All in all, comparison to the manual-based expansion, the system this saves roughly 70% of time. And however, I'd say reduced manual interactions are there. This means that you have less user influence and higher reproducibility, and this might even outweigh time-saving considerations at this point.
Thank you. We have one more question for you. Has the Prodigy system already been used in any clinical studies or cell therapy trials?
Yes, actually the system has been widely used for hematopoietic cells and more recently for CAR T-cell based clinical trials. And currently, according to our knowledge, more than 25 trials are underway using the CliniMACS Prodigy system worldwide. Also, we've learned a lot from cell therapies using hematopoietic stem cells and CAR T cells, and we now feel ready to transfer this knowledge to pluripotent stem cell-based therapies.
Thank you again, Sebastian and Chao, for your time today and your important research. Do you have any final comments for our audience?
Thanks a lot for everyone to join us. And like I said in the beginning, we really wish that these technologies would help you with your project. And if you are interested in our CliniMACS Prodigy Adherent Cell Culture System for automated and GMP-compliant stem cell manufacturing, please contact us or the Miltenyi local office, and we would like to provide more information for you. And also, live demos are also available on demand. Thanks a lot also for our host, LabRoots. Yeah, this is final remark from our side.
Thank you. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.